Integrative Gene Regulatory Network Inference using multi-omies data Neda Zarayeneh 1, Hun Oh 2, Donghyun Kim 3, Chunyu Liu 4, Jean Gao 5, Sang-Si Su 1, and Mingon Kang 3 1 Department of Computer Science, Texas A&M University-Commerce, Commerce, Texas, USA 2 Department of Medical Physics, Memorial Sloan Kettering Cancer Center, New York, USA 3 Department of Computer Science, Kennesaw State University, Marietta, Georgia, USA 4 Department of Psychiatry, University of Illinois at Chicago, Chicago, Illinois, USA 5 Department of Computer Science and Engineering, University of Texas at Arlington, Arlington, Texas, USA Correspondence, Wuhang 9 at Kennesaw.edu. Abstract Biological network inference is of importance to understand underlying biological mechanisms. Gene regulatory networks describe molecular interactions of complex biological processes. Graph models are mainly used for gene regulatory networks, where nodes and edges represent genes and their regulations respectively. In the most research, the molecular interactions, edges, of gene regulatory networks are inferred from a single type of genomic data, for example, gene expression data. However, gene expression is a product of sequential interactions of DNA sequence variations, single nucleotide polymorphism, copy number variation, histone modifications, transcription factor, DNA methylation, and many other factors. There are high-throughput genomic data that measure the various biological processes. We call the multiple types of genomics data as multi-omics data. In this paper, we propose an integrative gene regulatory network inference method, iGRN, that can incorporate multi-omics data and their interactions in the graph model of gene regulatory network. Copy number variation and DNA methylation were considered for multi-omics data in this paper. The proposed method, iGRN, was applied to the human brain data of psychiatric disorder. Through the experiments, iGRN showed its better performance on model representation and interpretation than other integrative methods in gene regulatory network inference. Keywords: Integrative Gene Regulatory Network Inference, Multi-omics data. Introduction. Gene regulatory networks, GRNs, provide comprehensive insight and in-depth understanding of complex biological processes. 1, 2. GRNs describe molecular interactions of complex biological processes by mainly using a graph model, where nodes and edges represent genes and regulations between genes, respectively. However, gene regulatory network inference is a challenging problem due to the large scale of gene expression microarray data. Moreover, the gene expression microarray data are typically noisy, and the number of available observations is much less than the potential interactions in the gene regulatory network. The importance of an integrative genomic study is being increasingly emphasized along with the rapid development of various types of high-throughput genomic data, since Human 978 150 90 16 10 5/16/$31.00 Copyright 2016 IEEE 1336 Diseases involve a sequence of complex interactions and multiple biological processes such as genetic, epigenetic, and transcriptional regulation. 3, 7. For example, gene expression is a product of sequential interactions of DNA sequence variations, single nucleotide polymorphism, SNP, copy number variation, CNV, histone modifications, transcription factor, DNA methylation, other genes in relevant pathways, and many other factors. 8, 12. Thus, SNP is not the only factor that can explain biological processes. CNV, which is a modified gene structure, often alters downstream pathways or regulatory networks. In a similar way, DNA methylation also often reduces gene expression in nearby gene by the methyl groups added to the DNA. For instance, monozygotic twins who have an identical DNA sequence often have different CNVs, which consequently cause discordance in congenital heart defects. 13, 14. Therefore, the multiple genomic factors such as multi-omics data should be integrated to have an in-depth understanding of mechanism in the gene regulatory network. The approaches of gene regulatory network inference are mainly threefold: 1. Correlation-based, 2. Bayesian-based, and 3. Regression-based approaches. Correlation-based approaches identify the interactions between genes by using linear correlation, for example, Pearson's correlation coefficient, on Boolean networks, which is the simplest model of gene regulatory networks. Boolean networks represent an active interaction as 1 and inactive as 0, and then active interactions of genes are determined if the correlation coefficient is higher than a certain threshold. WGCNA, weighted gene co-expression network analysis, is a representative method of the correlation-based approaches. 15. Along with the correlation coefficients, mutual information, MI, 16, maximum information correlation, MIC, 17, and conditional mutual information, CMI, 18, 19, have been also used on Boolean networks for gene regulatory network inference. However, the correlation-based approaches lack the interpretation of causal effect between genes since they use an undirected graph. Bayesian-based approaches for gene regulatory networks allow one to infer causation and causal relationship between genes based on a directed acyclic graph, DAG, in a probabilistic manner. 2023. Moreover, Bayesian network inference approaches can deal with a missing value problem, and can efficiently incorporate prior knowledge such as pathway database in the Bayesian model. 22-23. However, it limits the number of genes to infer the gene regulatory network, since the learning cost grows exponentially. Regression-based approaches infer the relationship of genes by decomposing the gene regulatory networks into p, number of genes, regression problems. 24-27. The regression-based approaches have often adapted lasso for providing biologically interpretable solutions on the decomposed regression models. Such approaches showed robust performance even with large-scale gene expression data. For instance, regression-based approaches were highly ranked as top-performing methods in the HPN Dream Network Inference Challenge 2. However, only limited research has been performed on the multi-omics data for gene regulatory network, due to the challenge of developing an efficient computational method that integrates these heterogeneous data into a standardized format. DCGRN proposed an integrative gene regulatory network inference method by using gene expression, CNV, and DNA methylation. 25. In DCGRN, CNV and DNA methylation were considered as independent components as well as genes. Therefore, it lacks the interpretation of interaction with genes in the model. In this paper, we propose a novel integrative gene regulatory network inference method, iGRN. The contributions of this study are: we infer a gene regulatory network by incorporating multi-omics data such as CNV, DNA methylation, and gene expression, while most gene regulatory network inference methods used a single platform of gene-expressed SciN. iGRN enables one to biologically interpret the integrative model of the multi-omics data by taking into account the interaction effects between the multi-omics data. We first define notations in section ELA. Then, iGRN is described in section 2b. In section 3, experimental results are demonstrated, where iGRN was applied to psychiatric disorder. 2. Methods. A notation. We describe the notations that we deal with throughout this paper. Let G denote a matrix of gene expression, GENXP, where N is the number of the patient samples and P is the number of genes in microarray data. A vector of gene expression at the ith gene is denoted as GEN, and GI is a matrix that contains other gene expressions than gene I, GIENXP1. The matrices of CNV and DNA methylation are denoted as C and D, respectively. We suppose that a CNV, or a DNA methylation, is associated to a gene if it is located around, upstream or downstream, the gene on the same chromosome. The CNV and DNA methylation associated to gene I are represented as CENXY and 1337 Figure 1, example of iGRN. The interactive effects of copy number variation, CNV, and DNA methylation, DM, are incorporated in the gene regulatory network model. DENXME respectively, where Vi and Me are the numbers of CNV and DNA methylation which are associated to gene I. The adjacency matrix of gene expression in a gene regulatory network is B, EPXP. We assume that there is no self-regulation, i.e., B equals 0 where I equals I, P. B integrative gene regulatory network inference. We aim to construct an adjacency matrix to infer a gene regulatory network from multi-omics data. We consider the interaction effects of CNVs and DNA methylations with the corresponding genes. In ERN, the integrative interactions are represented as shown in Figure 1. When a gene, for example, G1 in Figure 1, regulates another gene, G4, ERN shows the integrative interaction of the gene, GL, with a CNV, CNV2, and a DNA methylation, DM2, toward the target gene, G4. The integrative interactions between a gene I and a set of CNVs and DNA methylations belonging to the gene can be described by Fisher's interaction model, 1, where registered is an element-by-element multiplication. The equation explains differentially activated gene expression on variations of CNVs or DNA methylations. For instance, CNVs, which are more or less duplicated, may affect different express signs on the gene. Thus, gene expression cannot fully represent the genetic processes by itself without the consideration of interaction effects. The proposed Integrative Gene Regulatory Network is defined by incorporating interaction effects between multi-omics data. Genes have interactions with a collection of molecular regulators represented by the multi-omics data. Therefore, a legitimate hypothesis is that the expression of gene I can be represented by a sparse linear model of not only other genes but also CNVs and DNA methylations, which are associated to the gene I. Therefore, the gene expression, G, for gene I can be represented by G equals G underscore I plus, C registered GBF plus, D registered GBF plus A2, where B, BF, and BF are the coefficients of other gene expressions, CNV and DNA methylation of gene I, respectively. The residual is denoted as A. The adjacency matrix B of the gene regulatory network is comprised of B, 1, S, I, S, P, in, 2, IE, B equals, by, BT. The gene regulatory network can be inferred by optimizing the parameters. A learning function for the optimal parameters is obtained by using least squares with a sparse setting, argmax FB, GF, BF, ILGI, G underscore I plus, C0 GBF plus, D0 GBF 112 plus equal 1 plus equal full plus abfull, 3, where AG, AC, and AD are hyperparameters for sparsity regularization, and 1. 12 and 1 middle. 1 show L2 and LL norms, respectively. The optimization function can be considered as the following lasso problem, 4, where X is the augmented matrix, X equals GI, C0 G, D0 G. However, the number of genes in GI is much larger than of CNV and DNA methylation associated to gene I. For instance, there are only a couple of CNVs, C, or DNA methylations, D, for a gene in the psychiatric disorder data that we used for the experiment in the paper, whereas the number of gene expression in GI is hundreds even after preprocessing. Thus, the solution of lasso may tend to ignore most CNVs and DNA methylations despite their importance. Therefore, we solve the optimization problem in a stepwise manner. First, we identify significant genes that interact with gene I from GI by lasso, 5, then, a matrix of GI is constructed with the only genes of non-zero coefficients. Secondly, we compute p-values of the variables in the following linear regression, then, the coefficients of the genes, CNVs, and DNA methylations, where p-values less than 0.05, are assigned to the adjacency matrix, B. The procedure is described in Algorithm 1. Algorithm 1 iGRN 1, for IE, I, P, do2, B equals lasso, GI, G, in, 5, 3, compute the linear regression of 6, 4, BIG equals 0 if B equals 0 or p-value B less than 0.05 J BIG otherwise J5, BC equals 0 if B equals 0 or p-value BJ less than 0.05 JB otherwise 6, BD equals 0 if BTJ equals 0 or p-value BTJ less than 0.05 JBD otherwise J7, B equals BG8, and for 1338 10.07.5. J row greater than 0. 5.00. A 0. 2.5. 0.0 underscore L dot 0. Underscore 0.5, 0.0, 0.51. 0 log 2 disease / control figure 2, volcano plot for preprocessing. 3. Experiment results. A human brain data for psychiatric diseases we performed with human brain data consisting of psychiatric diseases and control on 131 samples. The multi-omics data of gene expression, CNV, and DNA methylation used in the preparation of this paper were obtained from the human prefrontal cortex, 28. The human brain data contains 39 samples of schizophrenia, 35 of bipolar disorder, 12 of major depression patients, and 44 of healthy control samples, where each sample has 25,833 of gene expression measurement, 1028 of CNV, and 24,399 of DNA methylathion. We considered the psychiatric disorder data as a group combining the three psychiatric disorder samples, since the psychiatric disorders share common features. For testing the reproducibility of the proposed method and its evaluation, we used two biological replicates of gene expression. By pre-processing from the 25,833 gene expression data, we used only 495 genes. The fold changes, FC, and p-values of the two groups, psychiatric diseases and healthy control, were computed, and the 495 genes, where their p-values less than 0.05 and FC less than 0.09 or FC greater than 1.1, were selected for further analysis, see Figure 2. We annotated CNVs and DNA methylations based on their locations. A CNV is annotated as associated to gene I if its region is overlapped with gene I in the same chromosome. DNA methylation is annotated as associated to gene I if its locus is located within 2 kbp around the transcription start site, TSS, of gene I. See comparison. We evaluated the proposed iGRN through experiments with two biological replicates of human brain data of psychiatric disorder. We compared the performance with ordinary regression-based gene regulatory network inference, GRN, and DCGRN, 25. Briefly introducing, GRN infers the relationship with other genes on gene I by 7, Table I, comparison with biological replicates. Replicate number 1 GRN iGRN RMSE 6.82 plus or minus 2.22 6.23 plus or minus 2.49 4.54 plus or minus 3.02 p-value, Wilcoxon 1.80 E, 52.30 E, 32 signed rank test with GRN, DCGRN, hash of significant CNV, 110 hash of significant OM, 12188 replicate number 2 GRN iGRN RMSE 7.23 plus or minus 2.25 6.17 plus or minus 2.66 4.11 plus or minus 2.90 p-value, Wilcoxon 2.61 E, 47.3.50 E, 27 signed rank test with GRN, iGRN, hash of significant CNV 13 hash of significant OM 7 153. Similarly, there exists neither CNV nor DNA methylation in DCGRN's model. 4. Conclusion. In this paper, we proposed an integrative gene regulatory network inference method, where multi-omics data and their interaction effects are integrated in the mathematical graph model. To evaluate the performance of the proposed integrative method, we compared RMSE and the numbers of significant CNVs and DNA methylations in our model with GRN and DCGRN. According to the experiments, the proposed method, iGRN, may provide a better model of representation and interpretation than others. And DCGRN does. The future direction of this work includes 1. Simulation, 8. Study, 2. More extensive experiments with various biolog iCal data, and 3. Evaluation with Gene Regulatory Network. We conducted the following 2 experiments. 1. Root databases. Firstly, it is often difficult to conduct simulation mean square error, RMSE, and 2. Counting the numbers of studies, in particular, for integrative methods in bioinfor statistically significant CNVs and DMs. First, we computed MATIX. Multi-omics data interact reciprocally in integrative RMSE between GI and their functions, CENT-GI, or gene regulatory networks, which makes it difficult to generate G_IBF for GRN synthetic multi-omics data. Therefore, we need to develop CENT-GI equals G_IBF plus CIP plus DIB for DCGRN, an efficient method to generate synthetic multi-omics data. G_IBF plus C greater than 9 GBF plus D greater than 9 GB for iGRN. Secondly, there are a number of publicly available multi-omics data such as TCGA, the Cancer Genome Atlas. The RMSEs of GRN, DCGRN, and iGRN were 6.82 plus or minus 2.22. https://cancergenome.nih.gov, although we applied the Pro 6.23 plus or minus 2.49 and 4.54 plus or minus 3.02 respectively on the replicate post method to psychiatric data in this paper. We will conduct number 1 and 7.23 plus or minus 2.25, 6.17 plus or minus 2.66, and 4.11 plus or minus 2.90 on the more intensive experiments with such data and analyze them. Replicate number 2, see Table I. The RMSE of the proposed method. Lastly, we will evaluate the method with publicly available iGRN show the best representation of gene expressions. Gene Regulatory Network databases. Since the evaluation of we also performed the Wilcoxon-signed rank test, which is the method in terms of biological context is challenging a non-parametric paired two-sided test to statistically assess, but of importance, experiments with publicly available gene the RMSE performance. iGRN produced the statistically SIG regulatory network databases are necessary. Significant improvement of RMSE compared with DCGRN in the experiments with both biological replicates, although DCGRN showed the better result than GRN. In Table I, the p-values of the Wilcoxon-signed rank test between iGRN and DCGRN are much less than 0.05, which means that the difference of RMSE between iGRN and DCGRN is statistically significant. Then, we compared the numbers of the CNVs and DNA methylations which are identified as statistically significant in the model. Since there is no CNV or DNA methylation in GRN, we compared with only DCGRN. In DCGRN, only 1 CNV and 12 DNA methylations were identified as significant in the network. Whereas, 10 CNVs and 188 DNA methylations of significance were detected in iGRN with replicate number 1. It may imply that iGRN can give better representation of the integrative mechanism of the gene regulatory network than DCGRN. The similar results were shown with replicate number 2. We introduced 2 discoveries from our experiments with iGRN. It shows that the gene expression of FKBP5 is regulated by genes of ZBTB16, MT2A, FYB, and PDK4, and the interaction effects with CNV of CNPI 1052 and DNA methylation of CG00862770. In DCGRN, the integrative effect with CNV or DNA methylation is not shown. As another example, the gene expression of SNRPN is regulated by gene of SNORD1L53, CNV of CNP12294, and DNA methylation of CG02171545. 1339 references, 1, L, each II, a review on the computational approaches for gene regulatory network construction, pages 55 to 65, 2014. 2, S. M. Hill et al., Inferring Causal Molecular Networks, Empirical Assessment through a Community-Based Effort. Nature Methods, Volume 13, Number 4, pages 310 to 8, 2016. 3, V. and Christensen et al., Principles and Methods of Integrative Genomic Analyses in Cancer, Nature Reviews Cancer, Number 5, pages 299 to 313, 2014. 4, W. Zhang, F. Li, and Elney, Integrating Multiple Omics Analysis for Microbial Biology, Application and Methodologies, pages 287 to 301, 2010. 5. R. Higdon, R. K. Earl, L. Stanberry, C. M. Hudak, E. Montague, E. Stewart, I. Janko, J. Schwanier, W. Brummel, N. Kolker, R. A. Bernier, and E. Kolker, The Promise of Multi-omics and Clinical Data Integration to Identify and Target Personalized Healthcare Approaches in Autism Spectrum Disorders. Omics, A Journal of Integrative Biology, Volume 19, Number 4, pages 197 to 208, 2015. 6. E. A. Reballer et al., Using Omics and Integrated Multi-omics Approaches to Guide Probiotic Selection to Mitigate Chytridiomycosis and Other Emerging Infectious Diseases, 2016. 7. K. Chisek, M. Krochmal, J. Klein, and H. Mishak, The Application of Multi-omics and Systems Biology to Identify Therapeutic Targets in Chronic Kidney Disease, Nephrology Dialysis Transplantation, p. GFV 364, 2015. 8. M. R. or et al. Individual and Combined Effects of DNA Methylation and Copy Number Alterations on miRNA Expression in Breast Tumors, Genome Biology, Number 11, p. R. 126, 2013. 9. J. R. Wagner et al., The Relationship Between DNA Methylation, Genetic and Expression Inter-Individual Variation in Untransformed Human Fibroblasts. Genome Biology, Volume 15, Number 2, p. R. 37, 2014. 10. M. Kang, D. C. Kim, C. Liu, and J. Gao, Multi-block discriminant analysis for integrative genomic study, Biomed Research International, Volume 2015. 11. Integrative approach for inference of gene regulatory networks using lasso-based random featuring application to psychiatric disorders, in Proceedings, 2015 IEEE International Conference on Bioinformatics and Biomedicine, IBM 2015, pages 145 to 150. L2, M. Kang, J. Park, D. C. Kim, A. Biswas, C. Liu, and J. Gao, Multi-block bipartite graph for integrative genomic analysis, iGRN Transactions on Computational Biology and Bioinformatics, Volume pp. Number 99, p. 1, 2016. 13. J. Breckpot et al., Differences in copy number variation between discordant monozygotic twins as a model for exploring chromosomal mosaicism in congenital heart defects, Molecular Syndromology, Volume 2, Number 2, pages 81 to 87, 2012. 14. C. and Henriksen, E. Chainat, and A. Raymond, Copy Number Variants, Diseases and Gene Expression, 2009. 15. P. Langfelder and S. Horvath, Vjicina, and R. Package for Weighted Correlation Network Analysis. BMC Bioinformatics, Volume 9, p. 559, 2008. L6, G. Altai and F. Emmert Stribe. 17. D. and Resh F. et al. L8, Gene Regulatory Network Reconstruction Using Conditional Mutual Information. Eurasian Journal on Bioinformatics and Systems Biology, Volume 2008, Number 1, p. 253894, 2008. L9, X. Zhang et I., Inferring Gene Regulatory Networks from Gene Express Cyan Data by Path Consistency Algorithm Based on Conditional Mutual Information. Bioinformatics, Oxford, England, Volume 28, Number 1, pages 98 to 104, 2012. 20. H. Nja and S. Jamasi, Weighted Ensemble Learning of Bayesian Network for Gene Regulatory Networks, Neurocomputing, Volume 150, No. PB, pages 404 to 416, 2015. 21. W. C. Young, A. E. Raftery, and K. Y. Young, Fast Bayesian Inference for Gene Regulatory Networks Using SCAN-BMA. BMC Systems Biology, Volume 8, Number 1, p. 47, 2014. 22. A. V. Worley and D. Husmayer, Reconstructing Gene Regulatory Networks with Bayesian Networks by Combining Expression Data with Multiple Sources of Prior Knowledge. Statistical Applications in Genetics and Molecular Biology, Volume 6, Number 1, p. Article 5, 2007. 23. D. Husmayer and A. B. Were 2. Bayesian Integration of Biological Prior Knowledge into the Reconstruction of Gene Regulatory Networks with Bayesian Networks. Computational Systems Bioinformatics I Life Sciences Society. Computational Systems Bioinformatics Conference, Volume 6, pages 85 to 95, 2007. 24. N. Omranian, 1. M. O. Alondombabi, B. Muller-Rober, and Z. Nikolowski, Gene Regulatory Network Inference Using Fused Lasso on Multiple Data Sets. Scientific Reports, Volume 6, p. 20533, 2016. 25. D. C. Kim et I., Integration of DNA Methylation, Copy Number Variation, and Gene Expression for Gene Regulatory Network Inference and Application to Psychiatric Disorders, 2014 IEEE International Conference on Bioinformatics and Bioengineering, pages 238 to 242, 2014. 26. J. H. O. and GeoDC, Inference of Radio-Responsive Gene Regulatory Networks Using the Graphical Lasso Algorithm. BMC Bioinformatics, Volume 15 Supple 7, No. Supple 7 p. S5, 2014. 27. M. Gustafson, M. Hornquist, and A. Lombardi, Constructing and Analyzing a Large-Scale Gene-to-Gene Regulatory Network Lasso-Constrained Inference and Biological Validation, iGRN Transactions on Computational Biology and Bioinformatics, Volume 2, Number 3, pages 254 to 261, 2005. 28. C. Liu et I., Whole Genome Association Mapping of Gene Expression in the Human Prefrontal Cortex, Mole Psychiatry, Volume 15, Number 8, pages 779 to 784, August